Hi everyone, welcome to topic 5. So for those of you that have been keeping up on your assignments with all of these changes in the dates and trying to be flexible, great job. Okay, so hopefully you're setting yourself up for success on the upcoming midterm exam. For those of you that still have assignments that are outstanding, try to take this week to catch up on everything because of the midterm exam coming up next week. So we want to make sure that you have done, learned all the concepts before you actually take the exam. So this is the week to really focus and catch up if you have fallen behind, okay? Before we get into our topic 5 assignments, I do want to mention that you have a couple of outstanding assignments that are going to be due this week. So if we go way back into topic 3, remember in the last assignment of topic 3, you did the experiment with the dialysis tubing, and basically you filled the tubes with different concentrations of sucrose to determine the rate of osmosis to see if there is an effect. So now you are expected to finalize your lab report on that experiment. So if you haven't had a chance to write your lab report, it is due on Sunday at midnight. And to help you, I have a couple of tips for you. So I have this instructor video that I created that walks you through everything that you need to include in your lab report. So this is a good starting point to help you, kind of piece together the different parts of your lab report. Dr. Blackwell, he also provided some videos about like what should be included in general for each of the sections. So this would be actually a better video to watch first, and then my instructor video that specifies the information for just this lab report next. And then once you have finalized your report, make sure, actually let me go ahead and switch this into student role. So give me just a second here. So that, because the reason being is that in your viewpoint, you actually have this grading criteria. So prior to submitting your lab report this Sunday, please go ahead and grade your report against the rubric here. That way you'll see right away if you accidentally left something out. Okay, so for example, a lot of students, they forget to include a title or their introduction is missing the hypothesis or something like that. So do take the time to go through and grade your paper against this rubric before you submit, because this is the same rubric I'm going to be using to grade your paper. So if you forgot to include a title, well, there it is, no points. Okay, so do grade your paper prior to submitting it so that you don't leave anything out. So our lab report is due this Sunday midnight. Okay, so try to get that in Sunday midnight. This is a 30-point assignment. So most of the assignments we've been doing so far are just worth 5 points. Actually, I think all the assignments we've been doing so far are worth 5 points. So this is worth 30 points. That's quite a bit. Okay, so try to do a good job on it. The other thing that is outstanding is your topic 4 quiz. Okay, so by now you should have taken a look at the topic 4, completed the topic 4 assignments, and hopefully, and then by today, everything will be graded so that you can go back and review what other students have posted and kind of compare your answers with theirs to make sure you're understanding the concepts completely. So that topic 4 quiz is due Sunday midnight as well. Okay. All right. And then again, if you forgot to submit an integration project topic, will randomly assign you one. So if you don't like the one that I assigned you, you can respond with one that you do like. Just make sure that it's one that no other student has chosen already. All right, so let's get into topic 5. So topic 5 is a continuation of what we were talking about in topic 4, where we're going to continue examining nucleic acids. And in this case, we're going to focus on how they play a role in protein synthesis, so making proteins. We will also learn how some genes that control whether the protein is made. Some of them, they will be expressed and others will remain silent. So we'll learn about that. And then we're also going to learn about how mutations in the DNA strand can alter the proteins, and we'll look at enzymes. So we'll see how enzymes play a key role in the body. So we'll see what kind of affects them. So in addition to our assignments this week, you will also be preparing for your midterm exam. Okay, so I have some practice sections or sessions for you that you can take a look at for this activity or these proctor sessions. Just realize that your exam for the midterm, it begins on the 14th. Yeah, the 14th through the 21st. So your midterm exam is available starting the 14th and through the 21st. I will hold proctor sessions for these midterm exams. So I have chosen 4 times that hopefully, at least one of these times will work with your schedule. So I am available for proctoring your exam on Friday from 2 to 4 next week, Saturday 3 to 5, the following Tuesday the 20th from 6 to 8 p.m. and then Wednesday 8 to 10 a.m. So I try to choose times that in the past, generally students seem to be more available in the afternoon and evening hours. And I did include a weekend time in there for you. So if none of these times work with you, however, you still need to have your midterm exam proctored. And so unfortunately, the CCC testing center won't allow online students or online classes to have their exams proctored there. So the only other option is ProctorU. So ProctorU is ProctorU.com is kind of an outside source. The problem is it does cost money to have your exam proctored through this outside source. So if you do decide to go the ProctorU route, sign up early to minimize the cost. I think the closer you sign up to the actual exam time you've chosen, the more expensive it can be because they have to find somebody that's available to proctor. And all of this is online. For your midterm exam, you don't come to campus for the proctoring, you just attend my Zoom meeting. Okay, so that way I can just, I will be there with you and any other student that's taking the exam at the same time. Every student will be in separate breakout rooms, and during the exam, you'll be asked to share your screen so that I can see that you are working through the exam without any extra resources, since it's a closed resource exam. And then I'm also there if something happens during the exam. So if your computer suddenly crashes or something like that, or if you have a question about one of the essay prompts or something that you don't understand something, then I'll be right there while you're taking the exam. Okay, so we have those 4 times available. So this week, take a look and see what works best with your schedule, and then you'll just sign up. Okay, so you'll just click the button. It's not open right now, so I can't click anything, but you can kind of see, you can choose one of these, and then you'll see, how many people have have already signed up. So generally, if there is a time where I end up with too many students, it's hard for me to keep track of everybody at the same time. So I might have to deny a student for that time frame. So try to sign up early for that. Okay. Let's go back. And then, oops, I don't like when it automatically signs me out. Okay, so as far as our assignments this week, we do have our topic 5 assignments that you'll be working through. So that includes the Adigia textbook reading as well as 5 assignments for this week. Yes. And then just be aware that you have, if you have your topic 4 quiz and your first final lab report due this Sunday, the 11th, okay? And then your topic 5 assignments, so T5S1 through T5S5 are due Monday. So it's a quick turnaround. Again, the reason for that is I want you to complete your assignments early so that you can take the rest of the week to prepare for the exam, this midterm exam. And the midterm exam is worth quite a few points. I can't remember off the top of my head. I think it's like 150 points or something big. So, you know, you want to do really well in the exam. And then there's just some other information if you want to look ahead. So as always, it's a good idea to take a look at Dr. Blackwell's summary video on topic 4, and he kind of introduces you to the information for topic 5. The reading assignments are topic 5 from your Adigia textbook. So do make sure you're keeping track online and completing your questions from the Adigia textbook. I go in every week and will update your grade based on what I see from your progress. Okay, so do make sure that you are keeping up with the reading. Your assignment student roles have changed. These are not us, okay? So ignore this. This is from a different section, different class. I haven't updated it just yet, but you know, make sure that you see which student number you are, and then that way you can keep track, just like always, of what you should be posting for the assignments. So let's take a look at our first assignment. So topic 5 S1, so transcription and translation assignment. So you're going to start out by watching this video. So this video is about RNAi, and you're going to basically use that information to help you kind of understand what's going on in the workbook. So let's take a look at the workbook. This is topic 5. So we always start with our preparation objectives that you might want to fill in from the Adigia reading. And then here we are, we're in topic 5. So the first part is pretty straightforward. You're just taking the information that you read about in the textbook and applying it. So, what is mRNA? What's the function of tRNA? What is siRNA and stuff like that. And then you're going to be comparing the DNA and RNA nucleotides. Okay, so here we have some cutouts for you. So you'll cut out each of these nucleotides, and remember, it is a nucleotide because it contains a sugar, a phosphate, and a nitrogenous base. So we're going to compare the nucleotides of DNA to RNA. And I can tell that this is DNA up here because it's deoxyribose sugar. RNA, it's ribose. Plus RNA has uracil as the nitrogenous base, whereas DNA has thymine, which is up here. Okay, so you'll cut those out and use this to help you kind of use these cutouts to help you kind of understand what each part of the nucleotide is and how they compare from a DNA nucleotide to an RNA nucleotide. And then you'll kind of fit the pieces together. So we have an RNA nucleotide and a DNA polymer. So you'll see that adenine is from RNA is only going to fit with thymine on DNA, okay? Cytosine on DNA will only fit with guanine on RNA. So it gives you kind of that complementary base pairing. And then we'll learn about transcription from your textbook and kind of understand how we're transcribing using a DNA strand and taking that strand and transcribing it onto an RNA molecule and how that occurs. Okay. So you get some practice with that. Here we have two template or transcription examples. So here we have notice this is the DNA strand, and you're going to be finding the complementary base pairs on the mRNA strand, so the RNA. So remember, RNA, there's no thymine present, so I should not see any T's in any of these boxes, okay? You see T's in the DNA, but not in the RNA because it's not present in RNA. So you'll figure out what's the complementary base pair. And for those of you that just looking ahead at your student assignments, you'll notice that we have example 2, example 1, example 2, example 1. That's just referring to this assignment. So were you example 1 or are you assigned example 2? So that's what you'll post, the mRNA for example 1 or example 2. And then the second part of this assignment, you're going to be looking at translation. So now that we have our mRNA strand, that's going to travel outside of the nucleus and bind with a ribosome in order to start producing a polypeptide sequence or a protein. So you'll learn about codons and anticodons in your textbook. So you'll apply that to our codon chart to figure out what the codons, what the correct amino acid sequence will be. So we have our mRNA, so that is in reference to this strand up here. So example 1, this mRNA strand will be copied, and then you'll use that strand along with the genetic code to translate the amino acid sequence. Okay, so if the strand in example 1 said something like AUG, then you'll write down what that means. It means met or start. Okay, in the next 3 lines, if it says CUU, you'll put LU, which means leucine, a type of amino acid, and you'll continue on. Okay, so that is the example 1. Example 2, you'll copy this strand down there, and then you'll do the same thing. So you'll have 2 times to practice. Okay, and then the video that was referenced in Moodle. Okay, so use this to help you kind of understand how, the overview of protein synthesis. So this can be a complex diagram. So just having kind of a video summary of, how transcription, translation occurs and all the different parts of protein synthesis. And then once you're done with that, it should be a little bit easier for you to answer the summary questions. Okay, so there's quite a few of them. So once you've completed your all of your answers, then you'll post your assigned answers into the discussion post. Okay. So this is due on Monday. Next, we have our lab summary assignment. So this is has to do with protein synthesis. So before you actually do the lab, you'll want to watch these videos. I gotta figure out why that one's not available. So what's a protein? What's mRNA translation? I'll try to figure out where the transcription one was and why it wasn't updated, because I think we updated it last term. I don't know why it's not in there. And then we also have like a little mini lecture on it. Okay, so once you've kind of understood the process, then you're going to complete the pages 61 through 65. So in this case, you're starting with a DNA strand, and you're going to transcribe it onto mRNA. And then you're going to take that mRNA strand and translate it into an amino acid sequence using the genetic code. Okay, so we'll start out with just the normal. This is a normal sequence, but then we'll see what happens when we have a mutation in the normal strand. So for example, here's our normal strand down there. It's the same as up here. But let's say that instead of a C, a T was there. Okay, so there was a mutation, a random mutation, that changed that C to a T. So what you're going to do is you're going to take this new strand, mutated DNA strand, and transcribe it onto mRNA and translate it into the amino acid sequence. Then you'll compare that amino acid sequence to the normal amino acid sequence and describe what you see. Okay, so what happened? You'll do and then you'll continue doing the same thing. So each time you're describing, okay, you're going to look at the new strand that was created, the new amino acid sequence that was created, and compare it to the normal. Okay, so each one of these, go back and compare it to the normal one. Okay, so you'll do that. We have 4 different mutations there. Okay, and then let's see, you're going to be submitting those pages for grading. Okay, so pages 161 to 165. So basically all of these mutations. Okay, so the best way to do that is to take your notebook and just take a picture of each page with your cell phone. And then I actually like the app Adobe Scan. So it's a free app that can actually you can just snap a picture in Adobe Scan and then flip the page, snap a picture, flip the page, snap a picture, and it'll actually convert it into a document, a PDF, and then that way you can just send it that way. So it's a lot easier than having to copy and paste things into different programs, or sending a bunch of photos that are kind of hard to read. So the Adobe Scan is just a useful tool. So it's by Adobe, and it's a free app. So check that out if it's makes sense for you to use it. Okay, so that again is due on the 12th. And then moving on, we have the 3rd assignment. So this is where we're going to get into the idea of gene regulation. And so we're going to learn how certain genes can be turned on and expressed, or certain genes can be silenced. And so to start that out, you're going to be watching this video, the Epigenome at a Glance. So you'll watch that video, and then you're going to look at this simulation in this Learn Genetics unit. So here we have this experiment that they did, where they learned about how certain genes can cause, if they're expressed, they'll cause one type of behavior. If they're not expressed, they'll cause a different type of behavior to be shown. So this video, you'll watch this video on the rats and kind of read through the information. Okay, so that way, once we've understood this whole process of this, lick your rat topic, then it'll help you kind of understand what's going on with the all of this information here. So let me take a look. So we're going back. So here we have. So again, once you've kind of read through watch the videos and read through the experiment, then it'll help you kind of understand, what is going on with gene editing and expression. Okay, so you can kind of see here we have a edit name for protein synthesis, and then here we have our gene expression. So the gene switches, how they get turned on and off, the involvement of histones and the methyl groups and so forth. And then this is the Lick Your Rats investigation, so you'll be using that activity to help you answer those questions. And then finally the summary of what happened and why. Okay. So once you have completed all of the questions, then go ahead and post your assigned questions in the discussion post. And then finally we get into enzymes. So far we've been talking about protein synthesis, so making proteins. So now we're going to talk about enzymes, which are generally proteins. Okay, so most enzymes are protein based. And so we have a video here that kind of explains how enzymes work and some of the things that can cause enzymes to denature. So you'll watch that animation, and then with the knowledge from your textbook that you've gained, you'll answer the question. So, kind of explain what's happening here with the enzyme is, this structure. So you can see that it doesn't change at all throughout the reaction. But we see the substrate is changing. Okay, so it started out looking like that, and now it looks like that as a product, and so forth. So you'll learn about, how that's involved with making like a polymer from two monomers or whatever subunits through dehydration synthesis. So something we learned about in topic 3. Here's hydrolysis. You learned about that in topic 3 as well. So here's basically using water to break apart the substrate. So enzymes being involved in that. Okay. So in this page 177, we're gonna just kind of take what materials you have in your house to help you understand this. So you will, if you don't have a dice, then use something else that's kind of similar size, and you're gonna scotch tape your fingers together, or tape them with, if you don't have scotch tape, masking tape, whatever. And then, try to see what happens, okay, when you're picking up objects. Right. And then here you can do the effects of, so this is an effect of, this is specificity. We also have cold temperature, so you can put, your fingers in ice water for a couple minutes, and then do the activity, see what happens. And then there's something where you will cross your fingers to see what would happen, as demonstrating the effects of heat or pH. And then we also have the mutation, so folding your thumb and 3 middle fingers together. Okay, how does that affect picking things up? So these are just basically showing how a change in your enzyme, in this case for our fingers, how that, if our fingers are, an enzyme here, small changes, how that affects how that enzyme functions. If there's a change in the way that, where the enzyme functions, then it's not going to be able to pick up the object as well. So that is kind of explaining what denaturing an enzyme and things like that, that role that it plays and how that happens. So once you've completed those activities and kind of wrote your observations down, then we have some summary questions there. Okay, and then you'll post the answers to your assigned questions in the discussion post. Okay. And then the final activity that you will be doing is an experiment. So the data from this experiment will be used in your second lab report. Okay, so the only thing you need to post in this discussion topic is the results of your experiment. Okay, so just be aware that you don't actually have to post your hypotheses or whatever it's discussed here. Okay. Prior to doing the experiment, you will need to prep your materials. So we're going to be making fresh enzymes. So to do that, you're going to be taking a potato, particularly if you don't have a russet potato, a different type of potato will work, but russet potatoes, they seem to work the best, extracting an enzyme and so forth. So you just basically cut it up and kind of blend it together, and then you kind of get this potato juice with water, and then you collect the potato juice from that and use that for your experiment. So in this experiment, we are going to be using that potato juice to see basically, so that's the enzyme. So the enzyme involved in this particular reaction. So the substrate that we're going to be using is hydrogen peroxide. So hydrogen peroxide, you can just get at the dollar store. You can get a whole container's worth. If you don't have it in a medicine cabinet, it's good for wounds and things like that. But hydrogen peroxide, what happens when it breaks down, it's H2O2, and it'll break down into water and oxygen gas. And so you're going to be doing this little experiment where you're going to be adding the enzyme to catalyze this reaction. Okay, so if we just had hydrogen peroxide, it'll eventually over time turn into water and oxygen, but by adding a catalyst, it'll speed it up. So the catalyst in this case would be the potato juice enzyme, or the enzyme that's in the potato juice. Okay, so you're going to be looking at what affects that rate. So we'll see how hydrogen peroxide converts or reacts to form those byproducts, and then what affects the enzyme's ability to do that. Okay, so we'll see how pH affects it, temperature, substrate availability, so that's the amount of hydrogen peroxide we started with, versus enzyme availability, so how much of the enzyme that we started with. Okay, so in this case, we're actually not going to be doing all of the experiments. So there's 4 different experiments here. You see there's a pH experiment, a temperature experiment, substrate and enzyme experiment. So for this online class, we're going to skip the enzyme or the pH experiment and the temperature experiment. Okay, so our class will only be working on these 2 experiments. Okay, the half of the class will be working on the substrate experiment. The other half of the class will be working on the concentration experiment. So you will only perform one of these 2 experiments. Okay, so to figure out which one you're performing, if you were assigned student 1, 3, or 5, you will be doing the substrate concentration experiment. If you were assigned the even number 2, 4, 6, then you will be doing the enzyme concentration experiment. Okay, so you only do one experiment, but there are 4 experiments that are being conducted. One of them's being done by other students, and then the other 2 experiments have already been completed and their data is posted here. Okay, so you don't necessarily need this data right now. You will be using this data in your lab report number 2, so we'll talk about that later. Okay, so for now, you're just reading the directions for your assigned experiment and completing it as such. Okay, so if you're doing the substrate experiment, then you're going to be using 4 test tubes, just like with the enzyme experiment. But each test tube will have a different amount of hydrogen peroxide added to it. But the amount of potato juice will be the same. For the enzyme concentration experiment, you'll have the same amount of hydrogen peroxide, but you're changing the amount of potato enzyme that gets added to the different test tubes. Okay? And basically what we're measuring is we're measuring how much the bubbles are produced. Okay, so you'll mix it gently and then you'll see all these bubbles or foam start producing. So that's indicating that the hydrogen peroxide is breaking down into water and then oxygen gas. So you'll see the oxygen gas being released, and you'll see it as basically the foam. So you'll measure how much foam is in the test tube, and that will be your results. Okay. So once you get your results, you're going to post them in your particular, assigned table here. So if you were the substrate group, then you're going to, add it there. Enzyme group, add it there. Okay. Once you post your data, then you'll take a look at what other students have posted and be able to add that information there as averages. Okay. So we'll deal with that later, but for now you'll just post your assigned experiment. Okay. And while you're in this topic, you might want to post the results of the pH and temperature experiment in the table on page 187. So here's the temperature, you can post what happened there, and then the pH, you can post what happened there. So you can fill out half of this table, just, so that you have it ready for when you go and actually do the lab report. Okay, but for this experiment, you're only posting your results for your part of the experiment in the discussion topic. Okay, so these are the 5 assignments that are due by Monday the 12th. Okay, once you have completed everything, then I highly suggest that you work on the studying for the midterm exam, but just note that there is an extra credit assignment that you can work on. That is just extra practice on basically looking at transcription and translation with a real application. So in this case, we're talking about cystic fibrosis. It's a genetic disease. And so you're going to be doing lots of transcribing and translating, and then comparing what happens with the different variants of cystic fibrosis. And so this is an extra credit assignment, and it is 5 points. So this is kind of extra practice. If you want to take advantage of that, you can do so, okay, and submit your answers. All right, so that's optional. And then you have your topic 5 quiz that is due on the 14th. So this is Valentine's Day, so make sure you complete that by then. So that way everything is ready to go for preparation for the midterm. Now, for those of you that are extra zealous and want to, start working on things, we do have the enzyme lab report. This is not due for a while. It's not due until February 26th, so you have quite a while to work on it. But it's basically taking the information from this lab, and you'll be using that to write the lab report. So if you looked at, for example, our lab here, you have an abstract, introduction, question, hypothesis. So you'll be constructing all of that, writing your method section, composing your results section. So I recommend this week, if you want to get a head start, is make a title and work through the introduction and method sections. Okay, and just get started on them if you can. All right, so let's see, anything else? I think I talked about everything. All right, so that's it for now. If you have any questions, feel free to email me. Again, we want to try to catch up this week, so if you have fallen behind, please make sure you're submitting those assignments sooner rather than later, so that everything will be, completed and understood, hopefully, before you take your exam. All right.